Okay, Suzuki Science Grade 12. We are here in week 2. Last week we were doing some evaluations. We ended off metabolism and we're starting a brand new unit. So in terms of how we're assessing you, at this point in terms of your grade, we have your first 2 units completed. We updated the marks. If there were any issues, hopefully you've contacted me already and we've had that conversation. And we are now moving on to the next part of the course. So this is a little odd in that your mark, whatever you have as your midterm mark, it will not go lower unless you choose not to do any work. So if you decide, you know what, I'm not going to do any work, I'm not going to show up, I'm not going to do anything, then the ministry, as I said this before last week, the ministry, the board, and myself, you will not get that mark. You do have to work. At the end of each unit, we're going to be giving you a task. And that task will be a part of the evaluation that we'll use to complement your grade as we work towards the end of the year. Normally in the past, we've done 3 units. We've done, 3 sections per unit. We did a test, we did a project of some sort, and we did a lab. The truth is, your enzyme lab was supposed to be the first half of the lab sequence. The second lab would have been a biotech lab, which clearly we're not doing any longer. In terms of your project, so what we'll probably do at the end of the unit, we will combine a project with some test questions, some multiple choice kind of questions. And the class is small enough that I could run it as an interview again. I'm still not exactly sure how we'll do it. We'll probably have some kind of presentation, PowerPoint, but that's something we're still working on. At this point, in terms of our weekly schedule, this week we're focusing on replication. That's the big idea. Next week we're focusing on central dogma. That's the big idea. And the week after that, we're focusing on genetic control, potentially including biotechnology. The 2 of them might go together. They might go separate weeks. If we decide to go with a 3-week or 4-week, most likely at the end of the 4th week is when your culminating task for molecular genetics will be due. After genetics, we'll be then moving into homeostasis, which is a big unit of study. We'll be spending 4 weeks on that. And then we'll probably spend 1 week on population. Once again, we're still not exactly sure what we're going to do in terms of your summative task at the end of the course, but that is something we're working on. We have to see how this technology and how the situation unfolds before we can really work on that. So this week on the screen, you can see that I have what it is that we're covering this week. It's a short week, right? Tuesday to Friday. In terms of concepts, it's really not that much in terms of what we've been doing in the past. So the first point is going to be DNA and scientists. And for the DNA and scientists, let me go over to that right now so you can see. There's a PowerPoint. I'm not going to teach this. I'm not going to do a voice cast. I'm going to focus on replication. So this PowerPoint is already online, and you can just go through it. And you are responsible for knowing a timeline of events. You should be able to start from sometime in the 1800s and move up to 2020, I guess. And you should be able to tell me the series of scientists who've contributed genetic knowledge to bring us to the point that we are at now. So it starts in 1865 with Mendel, which we talked about in grade 11 biology, and then each slide here talks about a different scientist. In most cases with these scientists, it will also either superficially or with maybe a little bit more detail talk about the experiment that they did that gave us more information. So the chromosomal theory of Boveri, the Griffith experiments, the Phoebus-Levine and what he contributed, McCarty-McLeod and what he contributed, Chargaff, Hershey and Chase, Wilkins, Rosalind Franklin is a very interesting historical story. I put a really big blurb here so you can read about it. Linus Pauling, whose model was completely wrong, but a lot of times it's the wrong things that help us move in the right direction. And of course, ending with James Watson and Francis Crick, who basically, most of the history books will tell us they're the ones responsible for telling us that the DNA is a double helix and figuring out what the actual structure looks like. As you go through this PowerPoint, you'll see a number of links. And so I encourage you to click on those links. I encourage you to click on these links and try them out. And they'll bring you to places where you'll either get YouTube videos or little activities that you can kind of work through the learning process that they did as well. So that's it. That's a scientist. Look at the PowerPoint, make a timeline, if I were doing a proper test, there would have definitely have been a timeline question asking you to tell me about the series of events that led to our modern double helix. The next concept is the DNA superstructure. So DNA, evidently you can stretch it to the Earth and the Moon like 6 times, or go around the planet like 25 times, or something silly like that. There's a lot of DNA inside your body. How is it possible to get that much DNA inside your body? So there's a whole series of structures. We start from the nucleotide. The nucleotide can be bonded with a sugar to form a nucleoside. That can be bonded with a phosphate to form a nucleotide. And you take a whole bunch of nucleotides together and you get a nucleic acid. So that is all stuff from biochemistry from the first unit. We covered that already. Then we get into some serious DNA structure, and you'll notice that I have it's superficial in this note. I don't expect you to go in all the detail. If you guys looked it up and you looked at some university text, or you went to some sophisticated animations, you'd see all sorts of really complex words and concepts. This, what I have on the screen here, is what you need to know. Okay, so you should understand how DNA is bound and compressed so we can fit Earth to the Moon 6 times and back and forth in our little tiny bodies compared to that distance I'm talking about. Okay, so you can use both these diagrams I have up here. They're both very similar, but the text is the same, and that is your DNA superstructure. The bulk of this lesson, the one where I'm going to teach, is in the next 2 points: DNA replication theories and DNA replication. For this lesson, I'm going to do the first 4 to get us started. And then this DNA sequencing is actually a biotech technique, but I want to embed it in this lesson. So that way, because it ties very nicely with what replication is about. So once you understand replication, then you'll be able to understand Sanger sequencing, and that will be one of the activities. So typically what we're going to do is we're going to give you the lesson at the beginning of the week. We talked about this already. On the Friday, at the end of the week, we'll give you some kind of, you'll tell us information about what you know, whether that's a test or a Padlet or Flipgrid, I don't know, something. And Wednesday will be the Wild Wednesdays. Some of you tuned in last week, and it really has not much to do with grade 12 bio, but it's something different. And then Thursday will be typically a game, or Tuesday is supposed to be the question of the week, but it's a weird week this week. So Thursday we'll either do a game or some activity or a question of the week. The DNA Sanger sequencing, and then in addition to all this, typically there'll be something you'll work on throughout the week. For this lesson, it's really just getting the knowledge of DNA replication, understanding the timeline of scientists, and learning the Sanger sequencing technique. That will be your learning for the week, and whatever product we decide to go with at the end of the week, we'll let you know a bit later.